All right, we are continuing talking about just kind of our basic introduction to the nervous system, and we're covering chapter 12 now, which talks a little bit more in detail about the central and peripheral branches of the nervous system. All right, so we're going to look a bit more at the actual gross anatomy of the central and peripheral nervous system, so this will tie in really closely with lab with a bit extra information thrown in there. Right? So we're going to start off in this video talking about the support and protection of the brain. This ties directly into what we're covering this week with meninges and ventricles and stuff like that. So this will be really, really helpful. All right, so the central nervous system obviously needs some protection, and it's going to be protected by a variety of different structures. The bones of the skull and vertebral column are kind of the obvious ones. Those are going to house the brain and spinal cord and offer a lot of that very hard structural support. Next up, we have the meninges. These are membranes made up of a lot of connective tissues. Depending on which layer of the meninges you're looking at, it's going to have a slightly different makeup of connective tissue, cells, and fibers. But these will help support, stabilize, and partition all of the nervous tissue. So it's helping to keep different nervous tissue separate from other structures. Next up is the blood-brain barrier. This forms what is called a privileged blood supply, and that just means that this is a specialized supply from the bloodstream that is ideally preventing toxins and pathogens from getting into the central nervous system. There are cases where things can still get past the blood-brain barrier. Meningitis is one example of those. So meningitis, by definition, is inflammation of the meninges. All right. And there's some different viruses and bacteria that can cause that. And then there are some toxins known to be able to cross the blood-brain barrier. But this is a very specialized blood supply to the brain that's going to help protect all of the central nervous system tissue from most pathogens and toxins. And then last but not least, we have the cerebrospinal fluid, or CSF. So this is cerebro, so the cerebrum, and cerebellum, so the brain, and then spinal for the spinal cord. This is going to circulate inside of and around the central nervous system, and this is going to help provide the nutrients that those cells need, remove waste products, and help cushion a little bit as well. So we talked about this a bit in a previous video with that choroid plexus. Those special cells are helping to filter out the blood to create cerebrospinal fluid. So here is a graphic showing those different layers. So you will see a lot of these terms in lab this week. So here we have the brain tissue right here. So this is the central white matter. This would be that cerebral cortex. And then immediately on the surface of the brain and spinal cord is the pia mater, or pia mater. I've heard it pronounced both ways. And that's this pink tissue right here that you can see. It is following all of the sulci and gyri of the brain. So it is directly on the surface of the brain. Above that, indicated over here, is what's called the subarachnoid space. So this is a space. It's going to have some cerebrospinal fluid in it, and as you can see, there's connective tissue. There's also blood vessels running through this subarachnoid space. All right, but that blood supply, again, it's not going to be able to cross that pia mater section of the meninges. All right, next, this kind of more purplish layer, this is the acronoid mater. So this is another layer of meninges. In real life, this is very, very clear. All right, it just kind of looks like a thin, clear film over the surface of the brain. Okay, and then above that is the dura mater, and this is going to be the most visible, easy to see one. It's also usually going to be a little bit more white colored tissue, and it's very, very tough. All right. And there are 2 layers of dura mater. You do not need to learn the 2 layers. All right. You just it helps to know that there are two layers because one of the lab structures that you're going to learn is this superior sagittal sinus. This is a big vein, basically, that runs through that longitudinal fissure at the top of the cerebrum, and it is sandwiched in between the two layers of the dura mater. All right, so that can be a little confusing on some of the models where it's going to look like maybe there's dura mater and then the superior sagittal sinus and then like the arachnoid mater, and that is not true. All of those white lines are still going to be dura mater. So on some of the models, you will not see acronoid, pia, and the subarachnoid space. All right, so let's talk a little bit more in detail about these. So the dura mater, this is anchored to the inner surface of the cranium, so the inside of the skull bones and vertebral cavity. The cranial dura mater has the periostal and meningeal layers, so those are those two layers, and they can form cranial dura sinuses and septa, so that's that superior sagittal sinus. The spinal layer of the dura mater just has one layer, so it's just one and not two. And so you don't have to worry about that once you get into the spinal column. And then the region in between the bones and the dura mater is called the epidural space. And we will see that in one of the spinal cord models. There's just one that has the vertebrae around it still. All right, so that's the epidural space. It's going to be full of some fatty tissue also. And then the region underneath the dura mater is the subdural space. And after the subdural space, we have that arachnoid mater. So this is a very thin fibrous tissue forming a loose sac around the central nervous system. So like I said, it's kind of a thin, clear membrane. And then below that is kind of a mesh-like substance called the arachnoid trabeculae, found in that subarachnoid space. So that's all of these little structures right here that you're seeing. And that's filled again by the cerebrospinal fluid. And then the pia mater is a very, very thin fibrous membrane following those superficial convolutions, so hills and valleys of the brain and spinal cord, so it will fit into all of the grooves and indentations. All right, so here's kind of another way of looking at that. Here's bone, so some of the bones of the skull. Here's that superior sagittal sinus. We've got the two layers of dura mater here in gray, that very thin subdural space. The arachnoid mater is here in that kind of orangish red. Subarachnoid space has all of those trabeculae in them. And then pia mater, again, in this gray color down here, or brownish color, lining directly on the surface of the brain and spinal cord, following all of the hills, valleys, grooves, etc. All right, so the cranial dura septa, so these are divisions, so think of septum in the nose, and then we have the interventricular septum in the heart, so this is something dividing, partitioning off different structures. So that dura mater, the meningeal layer, is going to extend down into the cranial cavity at 4 different locations, creating some partitions. These are the cranial dural septa. Sorry if you can hear my cat playing with paper in the background. She's being really loud. I apologize. So we have the falx cerebri. This goes through the midline and separates the two hemispheres of the cerebrum. Then we have the cerebelli, which separates the two hemispheres of the cerebellum that you are not learning in lab. You will learn the two halves of the cerebrum though. Then we have the tentorium cerebelli, separates the cerebellum from the cerebrum. All right, so separating those two portions of the brain. And then we have this diaphragma sellae, which surrounds the pituitary gland, because that's a really important structure. All right, but you're not learning any of these in lab, but you do want to be familiar with them for lecture material. All right, so here's kind of what that looks like. So here's that superior sagittal sinus again right here, runs through that longitudinal fissure of the cerebrum. And here we have that first section of that meningeal layer that's going to split the right and left hemispheres apart from each other. Here is the one splitting the cerebrum from the cerebellum. All right, and we can't really see the other ones in this particular view. And you can see there's a lot of other vessels coming through here, sinuses and stuff like that, but the only one you're learning in lab is the superior sagittal sinus. All right. Okay, let's talk a little bit about blood supply to the brain. So obviously we need blood going to the brain, and we need to take blood away from the brain. So the internal carotid arteries and the vertebral arteries end up becoming a structure called the Circle of Willis. And the idea here, the reason it's structured the way it is to have a lot of redundancy. So basically, there are a lot of different pathways the blood can take, which helps to ensure that even if a blockage occurs in one of the vessels, the brain is still going to get the oxygen and nutrients that it needs if we've got blockage through just one part of it. So that's what it means by maintaining perfusion. So this keeps blood supply going to the brain even if we have a blockage or narrowing in one section of it. Right, there can still be some issues resulting from that depending on how severe that blockage is, but we are still going to have that blood supply going to the brain. All right, so for venous supply, venous return, so blood is coming back into circulation through those dural sinuses. So the superior sagittal sinus is the one that you're learning in lab. It's draining into what's called the confluence of sinuses. And so coming back to this picture, we can see here is our superior sagittal sinus. You can see all of these other sinuses in here that you don't need to learn the names of, but they're all going to kind of come and meet up down here before draining through the jugular veins, and specifically the internal jugular veins. All right, so those go into mostly those internal jugular veins, which then drain down into the brachiocephalic veins. So this is what that Circle of Willis looks like. So here we see where we're indicating that internal carotid artery coming in from the right and left, all right? And then that's going to turn into the middle cerebral artery. That's going to kind of keep going out towards the outer edges of the brain, but then we've got the vertebral arteries, basilar artery right there. And so you can see there's a lot of different pathways that this blood flow can take to get to the various regions of the brain. So if we have like a little bit of a blockage, say here in this vertebral artery, we're still going to get blood coming into that basilar artery from that other vertebral artery, as well as the carotids. All right, so really important to maintain that blood supply to the brain. So here's another look at the dural sinuses and veins. So here's that confluence of sinuses back here starting. So we're kind of gathering all of those different veins together, the blood from them, to drain into the jugular veins. So here is the sigmoid sinus right here that will drain to the internal jugular. All right, so that's where all of that blood supply is going, so you can see all the arrows indicating where that blood is flowing to. All right, the CSF and the ventricular system. You will be learning the ventricles in lab. So again, cerebrospinal fluid is produced by that specialized layer of cells called the choroid plexus. They are lining the open spaces within the brain, which are called ventricles. And again, those are remnants of that neural tube from embryonic development. We have two lateral ventricles that drain through the interventricular foramina, which connect them into the third ventricle. The third ventricle is going to then move CSF into the cerebral aqueduct, and then into the fourth ventricle. And then the fourth ventricle is going to drain that CSF into the subarachnoid space, where it gets reabsorbed into the blood. And some of that CSF is also going to travel down the central canal into the spinal column. All right. So you were learning all of these ventricles, the interventricular foramen, cerebral aqueduct, subarachnoid space, and so you'll learn all of those in lab this week. So This can be kind of hard to visualize on a lot of models, but this is kind of what that looks like. We do have some models that kind of show the ventricles. It's called a negative space model. And so these are the lateral ventricles right here. They're these kind of very large C-shaped structures with a little tail. They used to be called the first and second ventricles, which is why we go from lateral to third and fourth without a first and second, is because the lateral ventricles were originally called the first and second. Not sure why they were renamed. I'm sure there's a logical reason. I just don't know what it is. One of these days I'll remember to look it up. And then here's that interventricular foramen, so that opening right there into the third ventricle, which is going to kind of sit in between the two halves of the thalamus. And then we travel down the cerebral aqueduct, which is right here, into the fourth ventricle, which as you can see is much, much smaller. So there's our fourth ventricle. Whoops. And then we go down that central canal through the spinal cord. Right, so hopefully that helps a little bit with lab material, because you will be learning all of these. Okay, so here is a diagram. All of these arrows in this case are not showing blood flow, but they are showing cerebrospinal fluid circulation through the brain. So this kind of reddish tissue right here, I keep clicking my mouse, I'm sorry. This is lining those ventricles again. That's the choroid plexus. So that's where that cerebrospinal fluid is coming out. So as blood flows past those cells, we're filtering the plasma out of the bloodstream. Not all of it, obviously, but some of it, and making that cerebrospinal fluid to get those nutrients to all of the neurons and glial cells. And so that CSF comes out of the choroid plexus in all of the different ventricles. And so it will just kind of flow. If we start in the lateral ventricles, it'll flow down that interventricular foramen into the third ventricle, picking up more CSF. Then it will flow down that cerebral aqueduct into the fourth ventricle. We've got more CSF being produced there, down that central canal through the spinal cord. And then it'll come back up the other side of the spinal cord right here in the other section of the central canal. And that CSF will surround the spinal cord as well, and that will all flow back up here and kind of travel around the outer sections of the brain in that subarachnoid space. And there are these acronoid granulation areas right here that are going to be where the CSF kind of returns back into that venous flow to return and drop off all of the waste products, get rid of those, pick up some fresh oxygen and stuff like that. And we will wrap up this video here and continue in the next one talking about the cerebrum.